Hey gang, I'm getting in. Sorry, it took a little bit to run lab it was hard to do with small group. All right, Cole, come on in. You could not very much, okay? All right, I know.
You're just me and Jack.
No, you and James, and then
Okay, sorry.
Michael's not. Everybody else is online, so.
Oh, yeah.
All right, somebody else just rang my doorbell. Who is ringing my doorbell? Oh, this is such fun today. All right. Anybody not here? All right, that was me ringing my doorbell. Excellent. All right, so first and foremost, I'm gonna get Q2 packets for those of you that are here.
I got it. I'll send you it.
All right, so second quarter packets are out. This one's yours, this one's Cole's. This is Lily, just reference as an example. So do you have your binder with you today, or did you leave it at home? You do have it? Grab it real quick, because you may want to put this stuff in it so that you don't lose it. If you are not here and you, why is that so low? That's so weird. That's a little better. Okay, if you're not here, you don't need to worry because your Q2 stuff you don't need for 2 more weeks, but I have it for you anyway.
Just put it behind all this other stuff.
So for today's, sorry guys, we're still scrambling after lab. So for today's lab, I'm gonna post something for everybody. Just give me a day or so to get it out. And it's not to be turned in. So it's just to observe for you guys that aren't here. All right, Cole, are you ready?
Right there.
All right, so we're gonna go ahead and start. So James, can you make sense of that? On the top is anything I was handing back to you. The only thing that may be confusing is there are new syllabus pages. So lesson 8 through lesson 17, you just pull those pages out of your binder and put the new ones in, because those new ones in have the exact homework information, whereas the ones in your syllabus just say TBA. Does that make sense? Clear as mud, not clear as mud. So yeah, so like if you look right, look here first. Top ones here. This is all stuff that I'm returning to. This right here. Put all of that in here, that's what we're about to do. And then, do that. There you go. And then you go to the, after you get that, and you go back to the front. And you just pull out. So when you guys pick up your packets, since you're not here, when you pick up your packets, can you see this? No. So everything that's lesson 8 through the back side is this where Q3 starts.
Lesson 9 is the first lesson.
Yeah, okay. So yes, that's correct. So if you go right here to the front, you find your syllabus, which should be right here. That's perfect. You just find the page that starts with lesson 8. Here. Well, you can out these. There we go. So we put out because this is all the Q2 and it just says like homework TBA. Whereas this says homework, there's your actual like should be there or something. That one's wrong, but the others are right. All right, there you go. All right, and it's on your page. Okay, guys, sorry, we need to start. So we're talking cell processes today. So in today's lab, I'm 10 feet from people, so I'm gonna pop this off. In today's lab, we did some looking at fermentation, which is one of the processes cells can use to produce energy, just like cellular respiration and photosynthesis. So let's go over cellular respiration. Now, there is a video today, I believe it's in your slideshow, that I will just make sure that you guys have the link to if you're not here, because we watched it at the beginning of lab with all the people who were here. So we don't need to watch it again in class. So chemical energy is stored in food molecules. Specifically, we're talking about sugars for the most part. So energy is released by the breaking of chemical bonds. You guys may remember this from chemistry last year. Energy is released by the breaking of chemical bonds, and it's often given off as heat. And that's exactly what happens with cellular respiration. It's given off as heat. That's why you're warm to the touch. So a calorie is how we measure heat. And it is enough energy to raise the temperature of 1 gram of water by 1 degree Celsius, right? You may also remember this from chemistry. This is kind of review stuff. So fat can store more energy than carbohydrates or proteins. So chemical energy in food, ATP synthesis occurs 24 hours per day. And it is, what am I doing here? Okay, the calories you consume only indirectly provide your body with the energy it needs. They actually provide the energy to produce the high energy bonds of the ATP molecules that then provide the power to your muscles and energy for your brain's electrical processes. And you can come in. When you take in fewer calories than you need in a given day to run these systems, the body turns to fat stores and to a lesser degree protein from existing muscle to convert carbon compounds into ATP through cellular respiration. All right, so here's what we're talking about. The calories you consume are in the form of carbohydrates or sugars. They're broken down into sugars in the end, right? And that is used to fuel the process that makes ATP. ATP is made from ADP, right? It goes from adenosine diphosphate to adenosine triphosphate. And then when you break the bond to take it back to ADP, you release energy. And you have to fuel every one of those reactions. And part of what is needed for those reactions are your sugar molecules. So those are the calories we're talking about, is the heat that's given off from those sugar molecules. So,
The 1,100 calories.
It should have 0, should be 1,000. Oh no, 1,100. Should have a small case C. Doesn't matter really, because it's like a title. So yeah, so when you read calories here, these are kilocalories. We just call them calories, but they're really kilocalories. So when you see, 400 calories, or like my kombucha is like what, 60 calories, 50 calories, it's really 50 kilocalories. So here are steps in cellular respiration. We have glycolysis, pyruvate oxidation, Krebs cycle, and oxidative phosphorylation. Aren't these fun words? This is a lovely diagram of how they all interact. And some of these things are going to occur in different places inside the cell. So some are going to occur in the mitochondria, and others are going to be outside the mitochondria. So in glycolysis, this process is anaerobic. This is your first step in cellular respiration. So what does anaerobic mean? Does anyone know what anaerobic means? What's aerobic mean?
Movement oriented.
It actually means oxygen, using oxygen. So yeah, so anaerobic would mean it doesn't require oxygen. And so this is taking place in basically cytoplasm. And you've got glucose, which has 6 carbons, and it breaks into 2 pyruvate molecules, which each have 3 carbons. All right. And then that breaking releases some ATP. Now, here's the thing about this process. It requires ATP to make more ATP. And in the video, there's the Amoeba Sisters video with the cellular respiration that I'll be sure to link that you guys can watch, because we already watched it earlier. It talks about how many ATP are produced and how, it's not, memorizing it is not necessarily the point. It's understanding the process. So then you have a transition reaction. This is oxidative decarboxylation. So anyway, this is when your pyruvate is converted to acetyl-CoA, and you lose some carbon dioxide here. So you know that part of part of the respiration process is the release of cellular respiration, is the release of carbon dioxide, right? So then you have this Krebs cycle process. So this happens now, we're inside the mitochondria, and this is oxidizing each acetyl-CoA to carbon dioxide. So we've released it, now we're bringing it back. And then this is when ATP is released, and this is where we start involving the NADH and FADH2 that you guys saw last week. And then there's this whole electron transport chain that happens inside, right? And this is literally moving an electron down from one chemical reaction to another. But the end product of all of this is you're putting water and sugar and oxygen, mostly your sugar and oxygen, those are your big things, in. And in the middle of all of the reaction that's going on, you've got one little ADP molecule that is going to get a phosphate group added to it, and it's going to become an ATP molecule. And then when that breaks, you get energy. And then you're going to release your carbon dioxide and your water as waste. So glycolysis, this is that first step again, right? So glycolysis is when you have your 6-carbon sugar, which we said before, undergoes a whole bunch of little chain reactions, ding, 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 and it gets converted into these 2 pyruvate, which are a 3-carbon molecule. So you go from 1 6-carbon to 2 3-carbon. And in these reactions, ATP is made and NADH plus is converted to NAD plus, sorry, is converted to NADH. And you guys who've done chemistry before can understand what this looks like and what this means. So basically there's an energy investment phase. So you see here, we're putting 2 ATP in and we're using those 2 ATP to form 4 ATP. So when I said you need ATP to make ATP, you have to invest energy at one phase and then it pays off in another phase. And this word here, glycolysis, if you need to remember what's happening here, glyco. Okay, so glyco is sugar, glucose or sugar derivative, whenever you hear that prefix, glyco. And then what is lysis? Do you guys remember what lysis is? That's a common one biologically, is splitting or breaking. So breaking a sugar. You take a 6-carbon sugar and you break it into 2 3-carbon sugars, right? Glycolysis, glycolysis. So the pyruvate oxidation part, and these get very picky. These are super involved and we're gonna brush over them. But what my high points I want you guys to understand is you have to put ATP in to get ATP out, and that ATP comes from ADP and returns to ADP, and that's when you get your energy released. So you got your pyruvate, and it goes from glycolysis into the mitochondrial matrix. So inside the mitochondria, the innermost compartment of the mitochondria, where it's converted into a 2-carbon molecule bound to coenzyme A. And this one we call acetyl-CoA. And carbon dioxide is released and NADH is generated. So all this is going on. So what's crazy to think is that every cell of the billions of cells in your bodies are doing this constantly, incessantly, repeatedly, as long as they're alive, as long as you're alive. Then you have the Krebs cycle part, or what we sometimes people call it citric acid cycle, because there is some citric acid in there. So your acetyl-CoA, you just made that, right? Made with the last step, combines with 4-carbon molecule and goes through a cycle of reactions. And in the end, you get regenerate 4-carbon molecule, and you have ATP, ADH, and FADH2. And that's when you're releasing your carbon dioxide. Okay, this is interesting history for you. So this guy right here, okay, this is called the Krebs cycle, right? All right, this dude is Sir Hans Krebs. He's a biochemist. So if it's Sir, then what do you know about him if he's got that as a title? He was knighted, but by whom? Like what country?
England.
Typically, yeah. Okay, so he's been knighted by the British, Sir Hans Krebs, who came up with the Krebs cycle. Okay, now interestingly, this guy here, he is General Hans Krebs, and he was a Nazi. Both same name. So also, Sir Hans Krebs, the biochemist, was Jewish, and he had to leave Germany for England because people like this guy took over and wouldn't let him practice medicine any longer because he was Jewish. So we have Hans Krebs, the Nazi, who kicked out Hans Krebs, the biochemist, and Hans Krebs, the biochemist, went on to England and now has this entire very important process named after him and was knighted. So he wins the game for sure. Easy for sure. So this is a model of the Krebs cycle. Everything we just discussed. All these things are constantly going on in there. You got your little NADH and NADH plus and FAD and FADH2, all this stuff. I want you to know that it has steps. I don't want you to memorize the entire chemical reaction within each step, but I want you to know that it has steps, right? All right, the next step is oxidative phosphorylation. All right, what the heck is that? So this is chemiosmosis or electron chain. This is the electron chain part that we talked about before. So you have NADH and FADH2, and they're made in other steps, right, in previous steps. And what they're going to do is they're going to deposit their electrons in this electron transport chain, this thing right here. All right, and it's going to, it's hard to describe, but they transport the electron down, they go back to their empty forms. So these guys turn back into NAD plus and FAD because they've dropped off their electrons basically. And then the electrons are going to bump down the chain. So that energy is used to pump protons out of this matrix inside of the mitochondria. So they're pumped out and then you get a gradient. So you got protons that want to flow back in because now you've got we talked about concentration gradients where you have a membrane and you've got a higher concentration on one side than the other side. And it's always wanting to balance out, right? So if they're pumped out with an active process, then they're going to want to flow back in. So as electrons move down the chain, energy is released and used to pump them out of the matrix, and that forms a gradient. Then the protons are going to flow back into the matrix through an enzyme called ATP synthase. Now, when you watch the Amoeba Sisters video, they talk about how ATP synthase is awesome because it's what makes ATP. And she makes a little point about like, if I could be any enzyme, I'd be ATP synthase. So at the end of the electron transport chain, oxygen is going to accept the electrons and take up protons, and then it's going to become water. Because if it's taking electrons and protons, if you've got one electron and one proton, what do you have? What element do you have?
So we're talking about
One electron and one proton, what element is that? That's hydrogen, right? Exactly. So it's going to take up these extra things, and you've already got oxygen. So now you're going to get hydrogen with it, and that's how you're getting water. So here's your overview. This is the part I want you to know. I don't want you to memorize all the chemical processes involved. I want you to know that it's a process that releases energy, right, from food in the presence of oxygen. Because remember, some of these processes don't require oxygen, like fermentation, which we talked about before. So glycolysis, pyruvate oxidation, Krebs cycle, electron transport chain. This is the part I want you to know. So let's look for a minute. All right. How about, which I forget which direction I did these in. Okay, let's try this one. I can't remember if I'm going to ask which I'm going to ask first. Let's try this. Requires oxygen. Which process requires oxygen? Aerobic is going to require oxygen. So now we'll find out which order I put these in. Yeah, I did put it in correctly. Okay, excellent. Okay, cool. Now, how about which evolved first? Do you suppose it's aerobic or anaerobic? Yeah, it would be anaerobic. And is the mitochondria involved in anaerobic or aerobic respiration? Aerobic. And then how about, and whoops, I clicked fermentation accidentally. So yes, fermentation is going to be involved. It's not involved in the mitochondria, right? So, oh no, what did I say? Oh, it's anaerobic. Okay, yeah. Fermentation is anaerobic and mitochondria is where aerobic respiration takes place. Well, some of it takes place outside, as you saw. And the electron transport chain, which one is that? Anaerobic or aerobic? It was aerobic. And a Krebs cycle. Now this is a tricky question. Is it aerobic or anaerobic, the cycle itself? Yeah, good deal. All right, so that gives you some idea. Now, obviously this whole process of fermentation is older, it's less sophisticated, right? So the yeast we used in lab today are fungi, and they can do both aerobic respiration and fermentation. So the ones that were at the surface of the test tube that had access to room air had access to oxygen, and they could undergo aerobic respiration. But the fluid at the bottom of the test tube that contained the yeast, which was undergoing fermentation and putting out carbon dioxide, could not get access to oxygen. So it had to undergo fermentation. And in doing so, when it put out carbon dioxide, it mixed with the water and we got carbolic acid, and we tested for it with a particular indicator. So this is the video I'm going to make sure you guys get a link to watch at home, because we've already seen it here today. So it's basically a really nice review of everything we just said that kind of puts it in a little neat and organized fashion. So there are other ways of making ATP from glucose without oxygen. So this would include anaerobic respiration and fermentation, which we just were talking about, right? So cellular respiration versus photosynthesis too. All right, so this is, what do we know about these two processes? Cellular respiration and photosynthesis. Chemically, what do we know about them?
One uses light and carbon.
Light and carbon dioxide,
Yeah,
right? How about formula wise? What do we know about them? Chemical formula wise, do you remember? Okay, well, you'll see in a minute, they're completely opposite processes. So the energy in photosynthesis and cellular respiration is going to flow in opposite directions because their chemical equations are complete opposites. So if you look at this cycle here, it's showing you the light is your energy source. It's coming in, right? And these guys are producing sugars that store energy. Okay, and then down here where you've got cellular respiration, you're taking sugars and you're breaking it down and you're putting out carbon dioxide and water. Well, that carbon dioxide and water is going into the photosynthesis process and taking in energy, making sugar, breaking down the sugar, releasing energy and carbon dioxide and water, and back around and back around and back around, because they are completely opposite. So here's the formula for photosynthesis, right? Everybody recognizes this one. So 6 what? Water.
6
Water plus,
6 carbon dioxide,
yep, with light energy yields what?
6 carbon. 12
What's this here? Do you know what that is? Or what kind of molecule? It's a sugar. Yep, sugar and oxygen, right? So when you say plants breathe out oxygen, this is what we mean, right? And then the opposite process, you guys are in the way. Here's the opposite process. So this is cellular respiration. So you have sugar and oxygen. In this case, your energy is going to be ATP, and you're going to release water and carbon dioxide. Because what do you breathe out? You breathe out carbon dioxide, right? Exactly. And water. You breathe out water too. All righty. So here is another diagram of how they all go together. And it's a good one to look at if you really want to sit and think about it for a minute. Like, how are they all interrelated? This is a good one to show how they're all interrelated. You're funny. So cellular respiration versus fermentation. All right, so this is like what we're doing in lab today. So anaerobic bacteria don't require oxygen for growth. So there are some bacteria, sometimes we're going to refer to them as archaebacteria or whatever, because they are the really, really, really old life forms, okay? And these are the ones that don't require oxygen for growth. So anaerobic cellular respiration is similar to aerobic respiration that in that electrons extracted from a fuel molecule are passed through an electron transport chain, right? Driving ATP synthesis. But some organisms use sulfate, which is this SO4 2-, right?
Yes.
As the final electron acceptor at the end of the transport chain, while others use nitrate, sulfur, or a variety of other molecules. So there are some very, very archaic life forms. That's where archaic is the same root as archaea or archaebacteria, that we'll call them nitrogen-based or sulfur-based life forms. These are the things you find like at the bottom of the ocean at deep sea vents. These are not the kind of things you find like giving you illness or living in the woods or anything like that. So fermentation. So what we did today was a fermentation lab. You get 2 main types of fermentation. Do you know what they are? Do you remember what they are from your reading? Looking.
Alcohol and lactic.
Alcohol and lactic acid. So alcoholic fermentation, that's what we did today in lab. All right. So this is, you get your NAD plus. So your glycolysis continues to produce ATP. However, alcoholic fermentation in yeast, for example, like today, produces ethyl alcohol as your waste product. And it also releases carbon dioxide, which is what we saw today, right? Because we were testing for carbolic acid being produced. And this is what happens in yeast, bread, beer, wine. So anytime you are using yeast in anything, it's alcoholic fermentation that's going on. So that's why, so when we make hard cider every year, we take the cider, the big jug of cider, and we put yeast in it. And really, you don't have to do much else. You just make sure everything's clean and nothing's growing in it. You put yeast in it and you walk away. And that yeast starts munching on all the sugar that's in the cider from the apples, right? It's just sucrose or whatever's in there. And it'll munch on it and it'll release carbon dioxide as gas. And then it will also produce alcohol as a byproduct, as the waste product. Quit texting. I know. But the more sugary the juice or the cider at the beginning, the more alcoholic the end product will be if you give it time, right? So the other form is lactic acid fermentation. So this one is a little different in that your NADH transfers its electrons directly to pyruvate and it generates lactate as a byproduct. So lactate, which is just the depronated form, so meaning it's missing a proton, of lactic acid, gives a process its name. So this is what happens with Lactobacillus acidophilus, which is the bacteria used to produce yogurt. Lactobacillus acidophilus. That's what's used to produce yogurt. Lactobacillus acidophilus.
Lactobacillus
Acidophilus. If you want to make yogurt.
Is that the big elephant thing
Yeah,
from
I know.
Dr. Seuss?
Oh.
Snuffleupagus?
No. Horton. I don't know. Is there one in Dr. Seuss? Snuffleupagus is Sesame Street. Mr. Snuffleupagus. Anyway, Lactobacillus acidophilus. If you want to make yogurt from milk or cream or whatever, you're going to need a culture of Lactobacillus acidophilus, and then you can make it. I know. But well, when you study microbiology, you can learn a lot from this. Like lacto would mean that it contains lactate molecule or produces it, or whatever. And then bacillus has to do with the shape of the actual protein itself. Like some are rod shaped, some are round, some are spiral, and that tells you about it. Now, the other place where lactic acid production takes place that is of impact to humans is in your muscles. So my two gymnasts in the room and anybody else that does athletics, you know that feeling when you have worked too hard and too much. So your muscles carry out lactic acid fermentation. Your muscle cells will. Though only when they have too little oxygen for anaerobic respiration to continue. So if you've been exercising really, really hard and you're panting, you ever get some people say that their muscles can feel crunchy, like kind of, crunchy. So it can happen. Yeah, you can produce lactic acid in your muscles, and lactic acid buildup in your muscles hurts. So you have to breathe a lot and drink a lot to flush it out. So again, you've got your glucose, which breaks down into pyruvate, and then these are your three main types of these are your products for fermentation. So there's aspergillus, which we didn't specifically mention, but it's a type of lactic acid fermentation, and lactobacillus, this will do it too. So these are the types of organisms that will undergo this process. So aspergillus is a type of bacteria, will undergo lactic acid fermentation to produce things like soy sauce. Lactobacillus will produce things like cheese and yogurt. And then these Saccharomyces, that's a hard one for me. Saccharomyces are the types of yeasts that will produce ethanol. Ethanol. Saccharomyces, yeah. Yeah. So
it's aspergillus.
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 Mm-hmm.
Lactobacillus.
Yep.
And
Saccharomyces.
Saccharomyces.
Yep. So in this case, if you look at so this is yeast, basically. Yeasts are this type of organism. All right. So if you look at carbon dioxide that's produced in bread, they're producing the carbon dioxide in bread, and that's how your bread gets all the little bubbles in it. Like that's how bread is full of air and bubbles. It's because the yeast has produced carbon dioxide as your waste product. In wine, for example, it has produced all of the, it's undergone all of the fermentation it can undergo. When you cork the wine up and ta-da, you get ethanol, just straight up ethanol. But in this case, it's CO2 and ethanol, and that's because it's still undergoing some fermentation process when you put it in the bottle or the can and put the cap on or seal it up. So it continues to undergo, but it's under pressure. So as soon as you open it, the dissolved carbon dioxide that's in the water within the beer is what suddenly bubbles and makes foam. Same way that happens with Coke, right? Exact same thing. Exact same thing. So here's a side by side, cellular respiration versus fermentation. Your steps are the same until you get to here. Your pathway becomes aerobic with cellular respiration. So you're involving oxygen and you're going to be spitting out CO2, H2O, and you've got your ATP. On this side, it goes anaerobic, and you're going to end up with something like lactic acid, ethyl alcohol, or other compounds. Right? Right. Now, the difference, I will say this, the difference between like wine and beer fermentation and spirits like vodka and whatever is that those are distilled, right? They go through the whole process where you basically you boil it and then you make a moonshine in the woods in a still because they're distilled, right? And you catch the vapors and the alcohol is in the vapors and then you reconcentrate it and bring it back down. All right, so photosynthesis. What's the definition of photosynthesis? It's going to take energy from where?
Sunlight and carbon dioxide in the air.
And yep, and it's going to store it as sugar. So it's the process by which plants capture light energy from sun and convert it into sugar. All right, cellular respiration. What happens here?
Cellular respiration?
You're going to take oxygen and sugar, right? So you produce ATP from oxygen in glucose typically is the sugar we're discussing. And you're going to release carbon dioxide and water. And then fermentation is going to be breakdown of sugars to make ATP in the absence of oxygen. So it's anaerobic. So I want you to know that you're getting ATP in both of these two processes, but this one is anaerobic. All right. So the formula for photosynthesis, as you guys know, is you get your carbon dioxide, your water, sunlight,
equals
6 sugars and 6 oxygens. Well, not 6, 1 sugar, 6 oxygens. Formula for cellular respiration is you're going to take 1 sugar and 6 oxygens, the same thing you produce here, and you're going to produce 6 carbon dioxide and water. And then fermentation, there's 2 different types, right? And it's different for each one. So where does photosynthesis occur? In plants. In what part? Where in the plant is it taking place?
Autotrophs. Right?
In autotrophs, yeah. But where within the plant is it taking place?
In the leaves.
In the leaves, inside the chloroplasts, right? In the inside the chloroplasts, on the thylakoid membrane, inside the grana, inside the chloroplast, inside the plant cell. Right. Okay.
Chloroplasts.
Huh?
I'll stick with
Chloroplasts, yeah. So where does cellular respiration occur?
Cells.
In specifically your mitochondria, right? Yes, this is the powerhouse of the cell because that's where you're producing your ATP. And fermentation, though, is going to be in the cytoplasm. So this is your main difference here about where it's produced. Your fermentation process does not take place inside the mitochondria. It's in the cytoplasm. So reactants and products, like we said, water and carbon dioxide yield glucose and oxygen. And then in this case, glucose and oxygen are going to yield water and carbon dioxide, exact opposites. Now here's where you can get this one. So glucose and ADP are going to give you lactic acid plus ATP, or you can get glucose plus ADP are going to give you lactic acid plus ethanol plus CO2 plus ATP. So these are your 2 main different fermentation processes. So How is cellular respiration related to photosynthesis? Well, how is photosynthesis related to cellular respiration? It's the same question, basically.
The reverse.
It's the opposite. Yes, absolutely perfect. And then, so what would be here would be the same answer,
Yeah,
right?
it's the opposite.
It's totally opposite. Yep. So then how is fermentation related to cellular respiration?
We all produce ATP.
Yep, they both produce ATP. That's the big one.
And
Yep,
Okay.
that's the big one. And it's anaerobic. And it is something all living things use both processes to release energy stored in food, though a lot of them are going to lean more towards one or the other. So what's the energy source for photosynthesis?
So,
The sun, right? And what's going to be the energy source for cellular respiration?
mitochondria.
Mitochondria is where it's going to take place, but the food is the energy source. So in this
Oh, they
case,
eat where?
glucose, sugar. So you have to have food in order to breathe, basically. That sounds weird, but it's true.
Oh,
And then what's the food source for fermentation?
so-
It's
that's
gonna
where
be
it
the same
occurs.
thing. It's gonna be the
Right.
same thing. Yep. Food, which in both cases is glucose. All right, so wait a minute. Whoop. What was that thing? All right, let's go back to that video one more time because I think it can be watched like 90 times and still would be good. So this is the video we watched earlier today. Actually, before we do that, let me just let you all know real quick where we're headed, those of you that are not here. So we're back from fall break. Ta-da, here we are, lesson 7. But you have one more lesson in this quarter, okay? And that's next week. And then you'll be having a test on unit 3, okay? And then please take note here where it says lesson 9, that you need to have any of your late work or whatever handed in, like anything left over from Q1, you need to make sure that that's done. Okay, there's a reminder about it, I think on your lesson for that day, but it needs to not be late. And then we're going to move into genetics and DNA. That's what's next. So now that we've wrapped up cells, basically, we're moving into genetics, DNA, Darwinism, heredity, heredity, can't say hard word. So next week's lab is a very easy one. Not next week's lab, sorry, week after next. Next week's lab is, which one is that? Oh yeah, this one should be fun. We need to get our critters in here. We're going to be using real live organisms next week.
Like what?
They haven't arrived yet, but planarians, which are little tiny flatworms. And it should be super, super cool. I'm very, very excited.
Are you surprised they get your testing?
No, they don't get in your intestines. I'll show you a picture of them. They're cool. And then we're going to do some modeling with some pop beads things. And then we're going to do this lab where if you were here last year, you extracted your own DNA. But this is going to be Shannon's favorite. This is the one where we're going to do DNA electrophoresis to find a DNA fingerprint for a criminal. And then we'll do protein synthesis labs and stuff. Okay, so back to this video. We'll do this video from today. Oh, Punnett squares. Yeah, we will be doing some Punnett squares. Let me show you what a planarian looks like since y'all don't know. So they look like this. Aren't they cute? They have little eye spots, which is kind of interesting. There's one. Oh, there's a diagram.
Are they microscopic?
Yeah, we'll be using our dissecting microscopes next week. Pretty cool. They're itty bitty. Yeah, we'll be using, like kind of like that. We'll be watching them regenerate body parts.
We're gonna chop their heads off? and watch them
We're gonna
regrow?
cut them in different directions and watch,
Ah,
they're gonna regenerate body
this is
parts.
decent. I'm not like I care about.
It's
Those
pretty
guys
cool.
I don't care about.
It's very cool. See, this is exactly what we're doing right
We
here.
have, like live iguanas and, like, cut the tails off.
Like here it says planarian regeneration. You know how some animals can regenerate, like starfish.
Question, how are we gonna cut this apart if they're microscopic?
You'll see.
These little exacto knives?
They have little tiny tools. So look at the lab. I'll show you what we're doing. Whoop, and we'll come back to that in just one second.
Yeah,
thing get tilted.
throw one of the lawnmower.
Oh my goodness. No,
Just
no
look it
lawnmower.
upside
That would
down.
make a giant mess of them.
Yeah, what do they come in? Do they come in like a bag of liquid or something?
They should come in a jar. I expect them in a couple days. I need to double check my order and see where they are.
Yeah.
So
Oh,
that's their diagram,
okay.
right?
Wait, go back. Why is their mouth down there?
That's where their mouth is. Yeah, no, it's
Do
weird.
they have eyes?
They have eye spots.
So
They
don't-
can sense light and dark, really. Can't do a whole lot
Where
of
do these
processing
guys
with that.
live? They live in
Water.
organisms?
They live in water. Mm-mm.
So what are the odds that we've swallowed one of these with something in a lake or
Oh,
something?
I don't know. Really don't know.
That's kind of gross.
So it's going to be we're talking about stem cells and cells that can regenerate. So going to put them in. So this is the type of cut you'll be doing. It's going to be very cool. And then watching to see whether or not they regenerate. So it should be pretty fun. And we're going to use our dissecting scopes. And as you can see, your tools involved will include paintbrushes to move them around, and you'll be using a scalpel with a brand new super sharp blade because you're going to need it to be very, very sharp.
Wow.
Yeah. All right. So we'll watch that video one more time if you guys are cool, because I think it's a good one and you could watch it 100 times and not ever see it too much. And then after this, we'll wrap up because we're getting close to done and there's nobody here. So actually, I don't know, James, if you want to text your mom and tell her she can come early, that's fine too. All righty, so here's this good old amoeba sisters.
Are you a morning person? One of us is, and one of us is definitely not. Mainly because when I wake up in the morning, it just takes a while for me to get my energy done. It takes a lot of time and coffee for that to happen for me. Cells really don't have that luxury. They are busy performing cell processes all the time. Active transport of many substances needed for their survival, for example. And the energy currency they need, specifically, is ATP. ATP stands for adenosine triphosphate. It's a type of nucleic acid, actually, and it is action-packed with 3 phosphates. We have a video all about ATP and how it works as an energy currency. So where am I going with this? Well, cells have to make this ATP. It doesn't really matter what kind of cell you are. Prokaryote or eukaryote, you have to make ATP. The process for making that ATP can be different, however, depending on that type of cell. One way is called aerobic cellular respiration. Lots of organisms can do aerobic cellular respiration, but this video is specifically going to go into aerobic cellular respiration in eukaryotic cells. That means this video is talking about the process within cells that have membrane-bound organelles, such as a nucleus and mitochondria. Eukaryotic cells include the cells you find in protists, fungi, animals, and plants. The mitochondria, which can be found in most eukaryotic cells, are going to be kind of a big deal in this aerobic cellular respiration because some of the process occurs in the mitochondria. So let's get started. Remember, the major goal for any organism performing this is to make ATP. Okay, here's the overall look at the equation for aerobic cellular respiration. Remember that reactants, inputs, are on the left side of the arrow, and products, outputs, are on the right side of the arrow. This equation, by the way, looks remarkably similar to photosynthesis. Look at how the reactants and products are on different sides. Well, that doesn't mean they're simply opposites. It does show that they have substances in common. In photosynthesis, organisms make glucose. Notice how glucose is a product. But in cellular respiration, organisms break the glucose down to make ATP. Fun fact, you know how when a bean seed first germinates in the ground, it isn't able to do photosynthesis yet? Yeah, the germinating bean is relying on glucose that it has stored, and it's doing cellular respiration to break it down to make ATP so it can grow. Of course, once it starts to mature and develop leaves, it can do photosynthesis. It will be able to do both photosynthesis and cellular respiration. Plants make glucose in photosynthesis and they break it down in cellular respiration. They can do both. But if you aren't photosynthetic, such as a human or an amoeba, you have to find a food source to get your glucose. You need glucose to get this process started, and we're going to assume that we're starting with one glucose molecule. Step 1, glycolysis. takes place in the cytoplasm, and this step does not require oxygen. It's considered anaerobic. In glycolysis, glucose, the sugar from the equation, is converted into a more usable form called pyruvate. Glycolysis usually takes a little ATP itself to start up. The net yield from this step is approximately 2 pyruvate and 2 ATP molecules, and 2 NADH. What is NADH? NADH is a coenzyme, and it has the ability to transfer electrons, which will be very useful in making even more ATP later on. We'll get to that in a minute. Now, an intermediate step occurs. The 2 pyruvate are transported by active transport into the mitochondria, specifically the mitochondrial matrix. In the mitochondria, pyruvate is oxidized. The 2 pyruvate are converted to 2-acetyl-CoA, which will be used by the next step. Carbon dioxide is released and 2 NADH are produced. Step 2, the Krebs cycle, also called the citric acid cycle, still in the mitochondrial matrix. The citric acid cycle is considered an aerobic process. While the cycle doesn't directly consume oxygen, some of the events in the cycle need oxygen to continue. To learn more about this intricate cycle where the 2-acetyl-CoA will enter, check out the further reading suggestions in the video details. Carbon dioxide is released. We also produce 2 ATP, 6 NADH, and 2 FADH. FADH is also a coenzyme, like NADH, and it will also assist in transferring electrons to make even more ATP. Step 3, the electron transport chain in chemiosmosis. This is just a beautiful thing, really. In eukaryotic cells, this is still in the mitochondria, and to be more specific, it's involving the inner mitochondrial membrane. We do require oxygen for this aerobic step. This is a very complex process, and we are greatly simplifying it by saying that electrons are transferred from the NADH and FADH to protein complexes and electron carriers. The electrons are used to generate a proton gradient, as protons are pumped across to the intermembrane space. All these protons being pumped out into this intermembrane space generates an electrical and chemical gradient. The thing is, if you remember from our cell transport video, ions, like the H+, don't easily travel across membranes directly without something to travel through. The protons can travel through an amazing enzyme called ATP synthase. If I could be any enzyme, I'd be ATP synthase because it has the ability to make ATP by adding a phosphate to ADP. ADP is a precursor to ATP. ADP has 2 phosphates, but if it obtains a 3rd phosphate, it becomes ATP. So in chemiosmosis, the protons travel down their electrochemical gradient through a portion of the ATP synthase, powering it to make ATP, the ultimate goal. Oxygen is the final acceptor of the electrons. When oxygen combines with 2 hydrogens, you get H2O, water. If you remember from our equation, water is a listed product. Now, the electron transport chain in chemiosmosis makes a lot more ATP compared to the other 2 previous steps. How much? So I've noticed in my years of teaching and the assortment of textbooks I've collected over the years, there are some different numbers in cellular respiration charts. In fact, you can even see them change a bit in different editions of the same book. And it's made me want to emphasize that it's important to not just memorize a number, because it really is more of a range. I want to focus less on the number of ATP made per glucose molecule because it depends on a lot of variables. One variable is the gradient we were talking about. How many protons were pumped across the mitochondrial membrane? You can see more variables discussed in the factual references, and those references have estimates ranging from 26 to 34 molecules of ATP per glucose molecule in the electron transport chain in chemiosmosis step alone. Then, if you add the other 2 steps, Krebs, otherwise known as the citric acid cycle, and glycolysis, you could estimate a range of anywhere between 30 to 38 net ATP molecules total per molecule of glucose. Again, to emphasize a range. Now, this was just one way of creating ATP, but like we had said at the beginning, all cells have to make ATP, but the way they do it can differ. If there's no oxygen available, some cells have the ability to perform a process known as fermentation. It's not as efficient, but you can still make ATP when there isn't oxygen. We have a video about that. We can't emphasize enough how important the process of making ATP is for cells. For example, cyanide, which is found in some rat poisons, can block a step in the electron transport chain, which would block ATP production. A poison that prevents ATP from being produced can be deadly. With the important role that mitochondria have in ATP production, there is also a demand for increased research on mitochondrial diseases. We are confident that the understanding of how to treat these diseases will continue to improve as more people like you ask questions. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
They have tons of great videos. I love them. Questions? You good? Okay, I'm gonna let this crew go. I'm going to put together some sort of presentation for the lab so that everybody knows what it is. It's not due to be turned in. You just want to be sure you understand what happened. And you guys could probably start on the homework and knock it out in the next few while you're waiting on
Is
your
it
mom.
online or is it in the binder?
I think it's online. All right, I'm gonna let this crew go. So we'll see you all later.
Yes.
Yep, bye. Bye, people.